In this video, I am going to teach you guys how to create a dihybrid cross. So far, all you have covered is monohybrid crosses, which are where you are looking at one gene or one trait and two different alleles for that trait. So for example, eye color or color of the seed, or whether the seed is wrinkled or round, rah-de-rah-de-rah, and they look like this. You guys are very good at these by now. However, unfortunately, genetics is not this simple, and now we need to introduce this concept of a dihybrid cross, where di in Latin means two. So instead of that, where we're just looking at one trait, we are in fact looking at two traits. So dihybrid crosses are to do with two genes, therefore two traits. Therefore, you can actually have a possible selection of 4 different alleles. Not to fear, I will explain how these things work. So I'm going to use an example from your SciPad. It's page 144, and what I would really like for you to do while you're watching this video is work along with me, take your notes. It may be a long-ish video, but if you're engaged and if you're taking those notes, then it won't feel long, it'll feel just like a breeze. So on page 144 of your SciPad, it is to do with floppy-eared rabbits. How cute. And what it tells you in the first little paragraph is that in rabbits, one gene controls coat color, while one gene controls the orientation of their ears. So let's write this out together. And I really want you guys to get in the habit of actually doing this when you are reading an exam question. So we've got gene number 1 codes for coat color, and it tells us that you can have a black coat, which is actually dominant and it is given a big B, or you can have a white coat. So remember, we're talking about bunnies here with their cute fluffy coats, and white coat is recessive, so we give it a little b. But because this is a dihybrid cross, we are comparing two different genes. So now we've got gene number 2, and this is to do with the orientation of ears. And the two different variations of that gene are oh gosh, I'll just choose random colors for this. Upright, which is dominant. So pointy uppy ears have got a big E allele, and then there's floppy ears, which is recessive, so it's got a little e. So there we go, 2 genes, 2 traits, 4 different variations of that trait, and that's what I mean here by 4 different alleles. Alrighty-o. So the question is, a rabbit breeder crossed a heterozygous black, upright-eared buck, male, with a pure breeding white, floppy-eared doe. Complete a Punnett square to show the expected genotypes of the offspring. Alrighty, so we have got a male, or in rabbit terms you would call it a buck, which is black with upright ears. So let's just do like a horrific drawing. Ears, oh my goodness. There we go, that's a rabbit, eh? And he's black. And he's got these upright ears. So what you should do is you should first of all write the genotype of this individual. The question has told us that he is heterozygous. So that means that he carries the dominant allele and the recessive allele, oops, I did ears second, I want to do coat first, and the recessive allele for both traits. So heterozygous for coat color and also heterozygous for the orientation of the ears. But his phenotype is displaying black and upright ears because they are the dominant traits. Our female, I'm going to do just over here, is pure breeding white with floppy ears. So again, another terrible drawing. And her genotype, she is pure breeding for white, and she's pure breeding for floppy ears. So she's homozygous recessive for both of those traits. Now what we have to do is we have to complete a Punnett square to show the expected genotypes of their offspring. And this is where we create our big boy Punnett square. It looks like this. 1, 2, 3, 4. These squares in the background are really useful for me. Alrighty, so this is our dihybrid Punnett square. I might just make a note there that this is their genotype of our two individuals. So what we've first of all got to figure out is what are the possible allele combinations that are going to be segregated into the gametes of these two bunnies. So remember meiosis, and remember that the last thing that creates genetic variation is the segregation of those alleles into those final gametic cells. So if we look at this Punnett square, we can actually imagine that these squares here are actually the gametes of that individual. So here's a little sperm cells, and here down this first column are the egg cells. And we've got to figure out what the combinations of alleles are that could be placed into those gametes. And the technique that we use for this is called FOIL. FOIL, I'll put it up here. And I do think that you actually use this in maths, maybe, FOIL. Right, so FOIL, F for first. What you do is you grab the first allele from each gene and you put them into the first gamete cell. So here, if we're doing the male, we grab the first allele from each gene, big B, big E, and we stick that in here. Big B, big E. O, we take the outermost alleles of that whole genotype and we stick that in the next allele. So O, we grab the outermost, so that would be big E and little e. I, we take the inner alleles, little b and big E. Little b, big E. And then L, we take the last allele from each trait or gene, so that would be little b and little e. Like so. So those are the possible allele combinations for those two traits that could be placed into the male gametes during segregation. Now we've got to do the same thing for the female. It's a little bit easier here. Let's do the same technique again. So I'll try to use the same colours. F for foil is going to be first, so we take the first alleles from each of those traits, little b, little e, so that could be going into one of her egg cells. Then we take the outermost, little b and little e. Then we take the innermost, little b, little e, and yep, that's the only potential allele arrangements that you can get. So it's all going to be little b, little e, by the way. But I'll just show you for the final. We take the last, which is B and E. Cool. So there we go. There's our possible allele combinations. Now what we've got to do is pretend and imagine what the genotype of the offspring are going to be if any of those gametes come into contact and actually get fertilized. So let's imagine that this sperm cell fertilizes this egg. What is the baby bunny zygote going to have in terms of its genotype? So we just put these together, and the two most important rules are that the alleles for the same trait have to go back together like this, and the dominant allele, if it's there, has to always be first before the recessive allele, like that. So there we go. I've put the two alleles back together for each trait, and the dominant allele has always gone in front of the recessive allele. Cool. So then you basically just go through and complete that whole Punnett square. And it may take me a little bit of time, but you are going to be completing this as well in your notes, because remember, you are following along with me. There we go, there's our completed dihybrid cross. And so all of these 16 squares in the middle here are showing us the potential genotypes of the offspring, if any of those gametes come into contact. So in an exam question, you might be asked to do that. And then you're also going to get asked, what is the expected phenotypic ratio? Expected phenotypic ratio of offspring. And you should be familiar with this from doing your monohybrid crosses. You will never be asked to tell the question what the expected genotypic ratio is, because there's so many different combinations, so don't even worry about that. You will have to give the expected phenotypic ratio. So what's the physical expression going to be of those genotypes? So we can see here, if we look in that first column, we have got 4 offspring that are going to be black with upright ears. We've got 4 offspring that are going to be black with floppy ears. We've got 4 offspring that are going to be white with upright ears. And we're going to have 4 offspring that are going to look just like their mum. They're going to be white with floppy ears. I'll just show you where I'm reading this from. This is my first column, black with upright ears. Don't want pink again. This is my second column, black with floppy ears. Third column, white with upright ears. And then finally, white with floppy ears. That is your phenotypic ratio, and that's exactly how you would have to write it as well, being super clear on what are they going to look like for both of those genes. Now, finally, I'm just going to end with the test cross. Test cross. We've done test crosses for monohybrids. You're familiar with it from level 1. You will probably get asked to do a test cross in an exam question for this as well. And this is something like a rabbit breeder believes that one of his males is a purebred black, upright-eared individual. So he thinks that the genotype of one of his male rabbits is this. But he doesn't know for sure because it could be this, couldn't it? The breeder doesn't actually know. So explain how the rabbit breeder would ensure that this rabbit was purebred and that you should complete at least 2 Punnett squares as part of your answer. So I'm actually not going to do the Punnett squares. I think you guys will be really good at that by the time you get to an exam question, but let me help you with the format of this answer. So to ensure that it's the buck, the male rabbit in question is purebred for black coat and upright ears, the breeder would have to breed it with, just the same as before with monohybrid crosses, the breeder would have to breed it with a homozygous recessive female. What's the female version of a rabbit? Doe. With a homozygous recessive doe for both traits, i.e. white and floppy ears. Please make sure that you're following along with me and taking these notes as well. If the offspring are all black with upright ears, the breeder will know that the buck was purebred. However, if any recessive traits are observed, the breeder will know that the buck must have carried recessive alleles. You guys know all this anyway. And that's where your Punnett square would come in really handy, where you would actually show, you know, like the pure, you know, if it was purebred, you'd do a Punnett square, and then if it was heterozygous, you would do a Punnett square as well, breeding it with that homozygous recessive doe. So there you go, there's your Punnett squares place, and I'm not going to do it for you. You could even do that now. You could pause the video and complete those 2 Punnett squares. Now, this is your excellence component that you have to have. Please do not forget this, because this is your guaranteed excellence at the end, that the breeder would have to carry out this test many times. So the breeder will have to carry out this test or breeding many times and produce many offspring to be sure of the Buck's genotype. This is because of the random / chance nature of fertilization and joining of alleles. There you go, girls. There's dihybrid crosses. Tons more examples to do in your SciPad. Have